Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is April 2nd, and we're talking to Dr. Daniel Llano from the Department of Molecular Integrative Physiology, University of Illinois, Urbana-Champaign. Dan is an auditory physiologist with an interest in the neural circuits within the chain of nuclei that make up the auditory system, that go from the ear to the cortex, I guess, is we normally think the auditory system ends in the cortex. And when sounds get there, we hear.
Sure.
Simple, but true in some sense.
By that.
And he's also a neurologist with an interest in aging and neurodegenerative diseases. So welcome, Dan.
Thanks for having me.
Also with us is Marina, Silveira, our local expert on auditory circuits, who you know if you're a regular here. Hi, Marina.
Hi, happy to be here.
And I'm Charlie Wilson. So I'm going to address both of you because I think the answer to this question might take both of you or eventually. So most of our listeners have an image of the auditory pathway starting at the cochlea and proceeding through a sequence of brainstem nuclei to the inferior colliculus, to the thalamus, and to the cortex, and ending in the cortex, as though sounds that make it to the cortex are finished being processed. Probably not quite true, but close enough.
Right.
But the brain structures at each level in this kind of intro to neuroscience diagram are boxes or shapes that might as well be boxes because they have no internal structure. And they have arrows flowing in, and the big arrows, the most important arrows, are going from the ear up toward the cortex. But there are also some other arrows that are pointing down from higher level structures to lower level structures. And these represent the connections among the nuclei. And then the nuclei themselves are usually just boxes. And which is just a revelation of our ignorance about them. But our ignorance is gradually disappearing. So one of the biggest and presumably the most important, because it's biggest and because it's obligatory in the pathway, is the inferior colliculus, but it's one of the ones that we know the least about. And so Dan and also Marina have both been working on trying to open that box and see what's in there, see what neurons are responsible for the arrows in both directions. So Dan, could you start us up maybe just by opening the box and tell us what the different pieces of inferior colliculus are and what's in there?
Sure. So I'm just going to call it the IC for this discussion. So most people, first of all, the IC, I've heard it recently described as being a jungle or an impenetrable jungle, huge structure. As you said, nearly obligatory relay for information en route from the auditory brainstem up through to the auditory thalamus and cortex. Metabolically, typically ranked as the highest or one of the highest metabolically active areas in the brain, that we don't understand that well at all. Typically, people will break it up into three main subdivisions: a lemniscal division known as the central nucleus, and then two non-lemniscal divisions, a lateral cortex and dorsal cortex. And there's various names have been out there for these structures, but the lateral cortex and dorsal, I would say, are pretty commonly used. And the lemniscal division is thought to be the main one that's sending information up the auditory chain into the ventral division of the metageniculate body, auditory thalamus.
It's named after a fiber pathway. Is that not right? The lemniscal one.
Yeah, right. I mean, so we use the term lemniscal primarily to denote the primary ascending pathways in this case in the auditory system, with tend to have short latency responses that are relatively sharply tuned to frequency, and tend to vary less to behavioral state than, say, the non-lemniscal pathways, which in this case would be the dorsal cortex and lateral cortex, which tend to have longer latencies, more complex tuning functions, and tend to be much more behaviorally variable in terms of their response properties. But those three nuclei are sort of what people have been working with for a while. Now, the other subnuclei have been defined as well over the years, but most people tend to focus on these three. But I would argue we still have a lot to learn about what even divides those three, right? I mean, defining their borders is not 100% agreed upon as to what those borders are. And then scratching beyond the surface at that point, it's anyone's guess in terms of the cell types. Been a lot of work done by Marina and Michael Roberts and other people looking at different cell types and what they might help us, how they might help us to define neural circuits in the IC. But I think we're still a long way to go to really understand what those cell types are and what the circuits are that they define.
So if there's a non-lemniscal part of the IC,
Yeah.
is it still part of the auditory system at that point?
Sure. I mean, so if you talk about, let's say, dorsal cortex and lateral cortex, these contain neurons that are acoustically responsive. They get inputs from the ascending stream of the auditory system. So if you put a retrograde tracer, for example, in the lateral cortex, you're going to label up um, uh, parts of the auditory brainstem, cochlear nucleus. You'll find inputs from the superior olive. But what you're going to find as well are massive inputs from cortical region, well, just from descending projections altogether. So from the cortex, from the thalamus, there's a projection, from the superior colliculus, and then of course from somatosensory areas, are going to be sending projections to the lateral cortex. So the lateral cortex, yeah, certainly I would call it part of the auditory system, but it's getting much more widespread inputs than the central nucleus.
So non-lemniscal is more a matter of inclusion, like what's included rather than what's excluded. It's not that the lemniscal axons don't go there; they do go there.
Yeah, absolutely. Yeah, right. That's a good way of putting it. So both lateral cortex and dorsal cortex, just based on the tracing studies, I mean, that we've done and previous groups have done, are getting ascending auditory input, but obviously quite a bit of other modulatory input that's altering their response properties, altering their tuning, and altering their roles and behavior.
And there's even a tonotopic organization in the shell. I see contradictory, but there is.
There is now, yeah.
It is tonotopically organized, is that right?
Right. So I mean, so the central nucleus we've known for decades is tonotopically organized. And the usual description of the non-lemniscal areas have been that they're non-tonotopically organized.
Yeah.
But now that, and part of that's because of just the way that the structure's been studied. I mean, traditionally it's been driving an electrode down into the IC, and the central nucleus, I mean, you pass through the tonotopically organized zones if you come at this at the appropriate angle from dorsolateral more towards ventromedial. And so the tonotopy in the dorsal cortex really wasn't described until we could image the surface of the dorsal cortex with 2-photon imaging. And so probably the most complete map that's been published has been by the Boris lab.
Yeah.
And they publish these beautiful maps, actually revealing two tonotopic maps, mirror image tonotopic maps on the surface of the IC.
Maps of frequency.
Yeah, maps of frequency.
Frequency.
Exactly right. And it's actually quite interesting because they show a reversal at the with the lower frequencies towards the middle, which is very different than the way. So the auditory cortex also has a tonotopic, or actually multiple tonotopic maps with a high frequency reversal. The dorsal cortex of the IC has a low frequency reversal, which I suspect likely has to do with the way that the afferents come into the IC in terms of their tonotopy being mapped upon the surface. So
this means that the two maps are touching low end to low end,
exactly,
or high
right,
to high,
low end to low end. So at this point, we have at least 3 tonotopic maps that have been described in the inferior colliculus. A large one that goes from dorsolateral to ventromedial through the central nucleus, and then 2 on the surface of the IC. They have this low frequency border that you mentioned, and we've looked for one in the lateral surface and haven't seen one. There's a fragmentary areas that look like they might be forming part of a tonotopic map, and we certainly haven't looked exhaustively yet. But at this point, there are at least 3.
Yeah.
So frequency is one of the things we expect to be mapped in the auditory system, but it's not the only thing.
Right.
I guess I would wonder about location in space and movement, detection of movement of
Right.
things, and
Right.
all of those things. Is the inferior colliculus interested? Are there any kind of maps
Yeah.
of those things?
Well, so in terms of amplitude modulation, I mean, that's been looked at quite extensively in the inferior colliculus. Amplitude modulation is a really important, feature for our ability to understand speech and other behaviorally relevant sounds. And the IC is the first place where you begin to find neurons that are tuned to particular AM frequencies. And there have been reports of mapping amplitude modulation, what's called periotopic mapping, that's orthogonal to the frequency maps. So that's been described, at least in the central nucleus. Space maps have been looked at extensively in mammalian preps, and I am not aware that anyone's been able to locate a convincing and reproducible space map in the IC at least. So how it is that we're able to localize sounds in space without a coherent and identified map. I don't know the answer to that question, Charlie.
So there's some in the superior olive. There's some spatial information must go somewhere.
Goes to the IC for sure. All that IC collects all that information from cochlear nucleus, various subnuclei of the superior olive, lateral lemniscus. So I don't have any doubt that the IC is extremely important in sound localization. It's just not forming an obvious 3D spatial map.
And also inheriting the information from the superior olive, right? So the main sound localization source will happen in the superior olive, and this information is inherited and processed by IC neurons. But if you damage the IC, would you have trouble localizing sound? I think that's the question, right? So contributes to maybe not the primary source of sound localization, but necessary for, I would say.
So in the visual system, there's a map of space and the visual cortex, where visual stimuli are. In the auditory cortex, is there no map of space telling where stimuli are?
No, I mean, the visual system, you have, the sensory world is already mapped upon the primary epithelium that receives visual information, right, on the back of the eye. In the auditory system, we're able to localize sounds, but again, as far as I'm aware, there isn't been a reproducible, generally accepted map of auditory space. I mean, whatever map we would have to be a computed map because it's not being directly inherited by the sensory epithelium. Those computations certainly take place, but maybe we don't need a map to be able to localize where sound is coming from in space.
Yeah, we're not really, we don't have a lot of models of how things get encoded. Like map is our favorite way of thinking about it.
Yeah.
Right? But if there's not a spatial map, there could be some other way of encoding stuff that we are not thinking about.
Yeah, I think maps are convenient tools, and they probably add, to computational efficiencies, right? But I don't know that every cognitive feat that we accomplish requires a map to be able to complete it.
Right. So in the box and arrow diagram, the inferior colliculus looks like it just passes on a signal up to the thalamus. And if you ask somebody, like if you're a student and you ask your teacher, why even have an inferior colliculus in that case? The teacher will say, oh, there's some important computation or processing happening there. We just don't know what it is, right? But we do know something about what it is, don't we?
I'm going to pass that difficult question right to Marina.
Yeah, we do know. We know some, right? Especially when we think about damaging the IC, right? So there's a lot of hypotheses of the IC being important for speech processing, right? So we have, for example, think of frequency tuning, and frequency discrimination. We have like those upwards and downwards FM sweeps. And there is a lot of hypotheses in the field that neurons in inferior colliculus, they respond best to either upward sweeps or downward sweeps, so which are extremely important for vocalizations, for example.
So these are like the sounds that come from the police and ambulance sirens. Right. They get whoop, whoop, like that.
Yeah.
And that's really an ideal sound for us to try to localize it, which is our task as a driver. And we hear one of those and we're trying to figure out where it's coming from. So that's there are neurons that are specifically sensitive to that kind of sound.
And the AM processing the IC as well is extremely important for speech processing. The IC is also a major source of plasticity, right, after hearing loss.
Sure.
So after there's like damage on the periphery, there's a lot of plasticity in the IC to compensate for that decrease in drive from the periphery to the brain. So
there are 3 different nuclei. Each one of them have a bunch of cell types.
a bunch
in
of cell
them.
types.
And the inputs end on some cell types and maybe not on others, and the outputs arise from some cell types and maybe not others. And we've also not talked about another important input, which is descending auditory information. So if I wanted to draw a better diagram, I would imagine my better diagram would have a little box inside the big box that is neurons of type A.
Yeah.
And they would have arrows coming into them, but the inputs would go to neurons of type A, and they would have an arrow going out to whatever type A neurons go to, and then there would be another one for type B neurons. So
It'd be a complex box.
it's going to be good. So that would make it normal for the brain, because it seems like they're always like that. So what do we know about the neurons that are the output of the inferior colliculus?
Yeah, so I 100% agree. If we could narrow down this jungle of cell types into the IC to some coherent classes, it would be extremely helpful for the field, right? Because as you said, we've got a box, right? We got arrows going in, arrows going out. But in the end, and we just look at what I would consider the progress that's been made, looking at the cerebral cortex, at cell types that exist there, or even in the basal ganglia, to help give us some guideposts as to how to interpret the data that we're finding. My reading of the literature from the inferior colliculus, however, is that there have been multiple attempts to identify cell types, and that when people do identify cell types or make an attempt to identify cell types, people chip away at it and realize that probably isn't a very good distinction. Even something really simple like GABAergic and glutamatergic cell types, they have multiple morphological types, right? We talk about disc-shaped cells and stellate-shaped cells in the IC, but both of those types can be either GABAergic or glutamatergic. Firing properties doesn't seem to help very much because those end up degenerating pretty quickly. In vivo response properties also degenerate pretty quickly. As I mentioned before, Marina and Michael Roberts, her former advisor, have made a lot of progress with various neuropeptides, and that might be a key. But I think that is going to be a major step once we have some cell types, and because we have the tools now to do all this really beautiful, sophisticated monosynaptic mapping, right? If we knew that, whatever marker it is you want to name identifies a coherent class of cells, we have the tools now to ask, what inputs do they get? Do they get inputs from, is it the contralateral IC? Is it from the dorsal-cochlear nucleus? What outputs do they project to? Because right now, as far as I know, most cell types that have been identified in the IC, if he asks, where do they project? Well, they project to the MGB, but some of them also project to the SC, and some of them project to the other
Back to the brain
side, and some of them project downward, etc. So I think that's, should be a major priority for the field. I almost feel that there should be, all the IC people should get together. Maybe we should do this and so forth, and come up with some coherent plan to be able to really, come up with a reasonable system. Because in the end, our lab is really interested in the descending systems that you mentioned. The descending systems to the IC are massive, right? I mentioned earlier they're heterogeneous. They're coming from layer 5 and layer 6. They probably outnumber the ascending systems.
Isn't that a surprising fact?
What's that?
That they outnumber the ascending.
Oh, yeah. And you find that certainly in the corticothalamic system, the same thing. But then if you were to ask me, and I've been working in this field for a while, well, what cell types do they land on?
Yeah.
I don't know. I could tell you regionally where they go, but I can't tell you more than that.
So the problem is, if you cannot define clear categories of cells, it's impossible to answer those questions.
Right. And then to complement that, we have the descending projections, but it's not only from cortex, right? We also have thalamic coming to the IC.
Mhm.
And one thing that I always talk to people in the lab, why would you need descending projection, which are primarily glutamatergic to the IC,
Mhm.
but then you have AC auditory cortex to thalamus, and then thalamus to the IC.
Yep.
They have these two sources of descending projections. Why?
Yeah.
Are they targeting different cell types? That's the only thing I can think of.
Yeah.
That the somehow some process is happening in the auditory thalamus to then send another descending projection, which somehow complement the cortical projection, but into different cell types.
Well, I mean, the thalamotectal pathway, this has sort of been my passion project, or I've been so interested in this pathway. It's evolutionarily ancient pathway where we see it in amphibians. It's probably their highest level of descending projections to the IC, and amphibians can do phenomenal tasks with their auditory system, right? They're amazing sound localizers. You can put a frog in a pond of thousands of other chorusing frogs, and it can identify its mate without any difficulties.
Nice.
It doesn't need a cerebral cortex to do that, right? Maybe this thalamotectal pathway has something to do with it. Again, it's the sort of the highest descending pathway that it has. And so somehow, mammals added this, corticotectal pathway or corticocollicular pathway on top of this pre-existing descending pathway. And as you said, now there's two sets of projections to the inferior colliculus, one evolutionarily old, one evolutionarily young. Do they interact, right? Does the corticothalamic pathway somehow synapse on neurons and then project to the IC, or are these completely independent pathways? So, these are just basic organizational questions about how descending projections get to the IC that don't even address the original question, which is about the cell types. So, a lot of super interesting experiments to be done, but yeah, I think we're a ways.
I think
ways
sometimes when things turn out to be hard, it's meaningful and it's a clue, and we should focus on it. So our illustrious predecessors used dendritic morphology to find cell types.
Mhm.
And it's true that there are modern techniques that have refined that, but the fundamental cell types they saw in the cortex and thalamus and brainstem and basal ganglia using dendritic morphology have held up.
Yeah.
They've just not been complete, and things have been added on. So the pioneers in the auditory system used this method, which worked pretty well in cochlear nucleus and in superior olive. But in inferior colliculus, it seems to have not worked. It isn't that they're different dendritic morphologies, it's just the dendritic morphologies don't seem to define functional cell
Exactly right.
types.
Yeah.
So, so what's that? What is it telling us? Like, is it possible that a cell that is functionally the same as that cell, but has its dendrites oriented this way, versus one this way, is like integrating two different things, but at the same frequency? Or are they really not? Are there really not dendritic morphology cell types, or should we have just abandoned it, quit looking at the dendrites at all?
Do you think it's like just a thought? It could be just the morphology that I see can be highly attached to the position of those cells on the synaptic axis, right?
Okay. Yeah.
So the stellate cells are the cells that extend their dendrites across the tonotopic axis, so they can be integrating different frequencies. By the disc shape, they have the dendrites with a narrow band of the tonotopic axis, so they are integrating within a single frequency. But GABAergic and glutamatergic cells can do that. So despite of the neurotransmitter that they are releasing, they still can respond, like integrate different frequencies of sound, not only respond, but integrating. So maybe the morphology in the IC is more related with this ability of neurons to either respond to a single frequency or integrate different frequencies rather than neurotransmitter that they will release GABA versus glutamate.
Great. And anyway, it's common
But I don't know.
for a whole bunch of different kinds of cells to release glutamate or GABA, and the basal ganglia, like in striatum, like all the cells.
And have different morphologies.
But they have different morphology and different functions. And the way you tell them apart is by their axonal projections. One group of neurons goes here, one goes there, or by the inputs they get. So you've done this work. You've like injected in various target structures. Can you summarize what you get?
Well, so I guess what I would say is it may very well be that there's just so many different cell types. Like, so within a class of morphologically defined cells, you may have so many subtypes within that, and those that project to this target or that target, and this neurotransmitter or another. That may be what we're being hung up on. We're focused on. Okay, so the cortex has pyramidal cells, VIP cells, SOM cells, and PV cells. Okay, and we're done. Well, I don't think it's going to boil down to 4 cell types in the IC. Maybe it's going to boil down to 30 cell types in the IC. I do think this idea of using projection-specific approaches. So the question is, like, what is the dimension to look at? Is it neuropeptides? Is it where they project to? And it is troubling to me that I feel like every cell type that's been identified seems to project to everywhere that the IC is known to project to. And it may be that the resolution of the techniques that we have is not really good enough to sort this out. So I'm attracted to barcoding techniques that allow you to be able to determine if an individual neuron is actually branching to multiple other targets, or to use, I think, more sophisticated techniques than what we've been using to try to identify the presence of branching and how frequent branching is. Because it's troubling to think that every IC neuron that's been identified projects to every target that's been identified.
Exactly.
We certainly haven't done the experiments, what I would consider to be the right way to really sort out branching. It's not really been a goal of ours, but that, I think, a very serious look at the branching patterns probably would pay off.
Yeah, correct. I think so too. I think that will really pay off.
So some of the, I mean, you sort of said GABA and glutamate doesn't help very much, but some of the structure can be seen that way. So both of you have looked at subsets of GABAergic neurons. One that forms these like morphological accumulations of GABA neuropil.
Right.
And then sparser regions in between.
Right.
And that's just in the lateral
cortex, right?
Called
Yeah.
external cortical. Yeah. So, what can we make of? I mean, that kind of thing. When we start seeing actual histological substructures, it's a big clue.
Yeah, I mean, that's one of the reasons we got so excited about identifying these modules. And I mean, the most important thing. So the work that was done by Alex Lasico in our group to identify that virtually every input or output from the lateral cortex can be defined by whether it's in or out of a module. So, for example,
it projects in.
Right. So the projections in as well as the projections out. So when we put retrograde tracer into targets of the lateral cortex, that retrograde tracer will end up either in the modules or in the matrix. So I do think the lateral cortex, because of that structural organization, gives us a toehold, right? I mean, you've heard me say before, I kind of look at the progress that was made in the striatum to say, well, okay, this structure kind of has that same general feel. Can we use the same approach that's been successful in other brain regions to understand the inputs and outputs of the lateral cortex? And so we're trying to do that. It would be more helpful if we had cell types, and a lot of the cell typing work that's been done really hasn't been done in the lateral cortex. Most of that work's been done in the central nucleus. But it would be fantastic if we could find, let's just say, NPY or VIP or CCK neurons, and if they were distributed in a particular way in the lateral cortex. We don't know that yet. We've not looked at that. That would be, a really useful tool to go along with the mapping. But for the time being, we're doing the mapping just, without those markers.
So which inputs go into these like islands of GABA, and which ones go into the sea in which they are floating?
Well, what ends up going into GABA modules for sure are somatosensory inputs, right? So the somatosensory inputs, so the lateral cortex of the IC has been long established to be a multimodal responsive area. When I say multimodal, I really mean somatosensory and auditory. And the somatosensory inputs that make their way into the lateral cortex are making their way into the modules. The auditory inputs are making their way, meaning like from auditory cortex and central nucleus, are coming into the C, the matrix around the modules. On the output side, a lot of that work has been less well defined, but the outputs to, for example, the dorsal cortex. So what's interesting about these non-lemniscal regions is that they communicate with each other quite extensively. And the outputs to the lateral cortex are coming from the extramodular space, from the matrix. And depending on where you're putting your retrograde tracer into the MGB, some of those inputs to the MGB are coming directly from the modules. But they're still, these are just pieces of the story that we have. What we really need to do, or somebody really needs to do, is to do this a little bit more extensively and put these retrograde tracers in different targets, you know, uh, all the known targets of the lateral cortex and work this out. But a handful that we have, basically everyone that we have looked at does map to either module or matrix.
What seems puzzling to me is that the designer of this nucleus would have put the somatosensory stuff in little globs
Yeah.
with auditory in between. I mean, you might think, well, let's just gather all the somatosensory up into one and the auditory up into another.
Yeah.
But there must be something spatial that they share. That's a reason why the somatosensory recipient thing ought to be near an auditory recipient thing.
Yeah. No, it's a great point. I mean, the modules, even though in a coronal section look like they're distinct, again, when they were reconstructed, so when Alex Lisico first established this organization, she did a 3D reconstruction of the modules, and they are elongated, right? If you look at them from a sagittal view, and towards the rostral end, towards the caudal end, they tend to link up, right? So, so they do certainly separate and spread across the inferior colliculus, but they aren't as distinct as they appear to be just by looking at the coronal sections. But yeah, your question of why do we need an auditory, matrix area next to a somatosensory module area? Why do we need to segregate these inputs at all? I mean, my hypothesis has been, there's so much convergence into the lateral cortex. There's at least 10 or 12 different brain regions that project there, that maybe you need some additional level of segregation to allow some processing to occur in the lateral cortex before somatosensory and auditory mix. And then clearly there is some mixing across the modules and matrix. And maybe that mixing is there to, suppress self-generated sounds, as it was discussed earlier. But that's probably the best, that I think we can, put together right now. But you need that spatial, adjacency to be able to allow that cross-modal suppression to occur.
Once the somatosensory information has made it into the inferior colliculus, is it going to get propagated up to the auditory thalamus?
Well, we're working on that, and I don't think those projections are going to the auditory thalamus. I think they may be going to somatosensory thalamus, and or to areas that are a little bit medial to the auditory thalamus that tend to have multisensory processing property, even the medial division of the auditory thalamus. I don't think it's going strongly to the lemniscal, ventral division of the MGB. But the areas adjacent to it and somatosensory thalamus are probably getting some of those inputs.
Yeah. So, and this is just one little piece. This is just this lateral cortical region that has this structure. And the dorsal cortical region, which is also receives non-lemniscal input, but shows none of this structure in it.
Right. As far as we could tell, correct. It doesn't have any of the I mean, so
does it have somatosensory responses?
Well, we've not looked. I don't remember what other people have seen. There aren't significant on an anatomical basis. We're not seeing inputs there. Certainly, the dorsal cortex of the IC is getting just proportionately getting more significant descending input from the auditory cortex than the lateral cortex. I mean, if we were to pick an area of the IC that gets the most, let's just say, of the total of all the inputs that it gets, what proportion are coming from the auditory cortex? that number is largest for the dorsal cortex of the IC. So it is an area that gets a lot of cortical input. It's likely going to be very important. It's probably going to be likely highly modulatable based on behavioral state. It also happens to be fairly, it's something that you can study now with 2-photon imaging because it's sitting right on the surface of the IC. It's easier to access than the lateral cortex. So I think we're going to learn a lot about it. I mean, there's multiple groups right now that are studying the dorsal cortex of the IC using 2-photon imaging. I mentioned Boris group and Pierre Fossilides group in Michigan. So I think that we'll learn more over the next few years.
Tell us a little bit about this because, we need a helping hand from some breakthrough technology to solve some of these problems. And this one seems like something.
Yeah, I mean, 2-photon imaging obviously has revolutionized the way that we understand cortical circuits. I don't know, maybe in the past 7 or 8 years, people have been applying 2-photon imaging to study the inferior colliculus. It so happens in a mouse that the inferior colliculus is accessible. It's sitting right on the surface. It's a little bit tricky because it's not, there's a large space between the skull and the surface of the inferior colliculus. There's a big venous sinus that's directly anterior to it, so that makes it a little bit tricky as well. But once the technical issues are dealt with, you've got this structure basically that is quite accessible for imaging. And in fact, you can probably image the two sides simultaneously and learn quite a bit about how. One thing about the IC compared to other auditory areas is probably the most bilaterally connected structure of the central auditory system. That's probably an arguable point, but there certainly is lots of connectivity between the two sides. So I suspect as more groups are, interested in this problem, have the technology to study this problem, we're going to learn a lot more about at least the surface of the IC. I mean, again, 2-photon imaging is only getting you the upper, portions, but it's getting us a lot more than what we've had in the past.
So this is using calcium imaging to try to cell by cell map electrical signals.
Correct. I mean, as it stands, the tonotopy on the surface of the IC, I mean, again, 10 years ago, nobody thought there was tonotopy, and now we see these really gorgeous maps. Whether there are maps for other things, like you mentioned earlier, amplitude modulated stimuli or frequency modulated stimuli or natural calls, all that kind of work is being done right now.
Yeah, so that's just immediately available things to do.
Yeah. I think these are all, I mean, there's enough groups that are actively engaged in this problem. I think we're going to learn a lot more about the dorsal cortex of the IC than the lateral cortex just because of the difficulty in accessing the lateral cortex. I mean, you can see a little bit of the lateral cortex as it kind of, curves around the top of the IC, but 80% of it is hidden in a sulcus. We've accessed that with a prism, but it's still, not, it's still challenging to do those experiments.
And you can do that in awake. I can't quite picture how that's done, but it must be a head fixed but awake animal.
Yeah, head fixed animal. The prism can be inserted, and you can do the imaging in an awake head fixed animal.
And so this removes other constraints that have always bothered people, that there are only certain kinds of situations in which you can test for sound responses, and the responses to sound of the animal change depending on state and what they're doing. And so likely they're going to change in the neurons as well.
Mm-hmm.
And
like anesthesia, right?
Yeah, anesthesia is great because it gives you a defined state to work with, but it's such a restrictive state that the animal wouldn't normally ever be in.
Right.
In that. So I'm this is how we reverse engineer the inferior colliculus. It's just like one step at a time, figuring out what all the components are and what all the wires are. But when that's done, because the inferior colliculus never operates independently of the rest of the network, then there's the problem of integrating our reverse engineered nuclei at every level with each other to reform an understanding of the network. And that seems really challenging to me because of all of these loops So do you have any like vision of that or any image of what that's going to look like or how that will proceed when we get to that point?
I'm looking at you for the tough questions.
Yeah, I think we'll get there, right? Because now we have the tools for it. It's not gonna be immediately, because to understand those projections, we're gonna have to, for example, have markers for each of the cell types that we identify. So let's say we identify 30, right? So how does 30 cell types communicate with the circuits? That'll be 30 different experiments using 30 different markers for one nuclei. So which one of those 30 cell types receive those ascending inputs? Which one of those 30 cell types receive ascending inputs? Which one converge and receive both? So it will be a complex question that is, I don't believe is going to be answered by a single lab, but rather than by a contribution from the field, from people studying different cell types and look at different projections, and eventually we can understand that.
And these loops can be opened up because even just momentarily with optogenetics or
Yeah, optogenetics would be
you
the
can
key to study
test
that.
with or without
Yeah.
some back projection.
Yeah, optogenetics will be the key, and now we can even test two nuclei within the same sample, right? So
Okay, well, I'm looking forward to that. We'll meet again in 2 weeks. I've made much progress. So
yeah,
thanks a
that was
lot,
fun.
Dan, for joining
Thank you
us.
very much for having me.
Thank you,
Marina,
Charlie.
and
That
It's
was
been
fun.
fun.
This has been Neuroscientists Talk Shop.